cellular respiration. This was chapter 3. Okay, this was basically the steps of using our biomolecules to harness energy. And the main biomolecule we talked about was glucose, right? So basically harnessing energy in the form of ATP from one molecule of glucose and then using that energy for cellular work. Okay, so we talked about glycolysis. And the important things we said about glycolysis is that it occurs in the cytosol. All right, here we make 2 net ATP. We also make NADH plus carrying electrons, right? Some of these electron carriers. We make pyruvate. And this is an example of substrate level phosphorylation. Okay. We actually make 2 pyruvate. So let me clarify that. So per glucose, we make 2 pyruvate here. Great question. Do we need to memorize or know the exact reaction of cellular respiration? So you don't need to know the individual steps of glycolysis, for example, of the linking step. You don't need to know the enzymatic reactions or even the enzymes. You just need to know the overall substrates and overall products, right? And then where it occurs is important as well. And then the mechanism by which ATP is made, right? So for example, glycolysis uses substrate level phosphorylation, which is borrowing the phosphate as the means of making that ATP. Then we had the linking step. This occurred in the mitochondria. Here we basically made acetyl-CoA and carbon dioxide, okay? But we basically convert the pyruvate that we get here into acetyl-CoA. Then the Krebs cycle, which is the next step in the process here, the Krebs cycle. This also occurs in the mitochondria, right? Here we make 2 ATP, some more of our carriers, right? We have FADH2, and then obviously some byproducts, carbon dioxide. And then the last step, which is the most important step, is the electron transport chain. This occurs specifically across the inner mitochondrial membrane. All right. Here we basically make 34 out of 38 ATP. We also said here that oxygen is required to be that final electron acceptor. Very, very important, right? Oxygen is required to be the final electron acceptor. Okay. Second C, right? So the final electron acceptor. The other thing we said here is that this is made by oxidative phosphorylation, right? So the ATP made by oxidative phosphorylation. Oops. So really important to distinguish where substrate level phosphorylation is versus oxidative phosphorylation, okay? So this is basically glucose oxidation and the stages that are involved and where we make ATP, right? We make some ATP here, two net here, so that's 4 out of our 38, and then the other 34 are in the electron transport chain. Okay, we also want to know that it happens in the cytosol for glycolysis, and then the linking step and Krebs cycle are in the mitochondria, and then we've got the electron transport chain across the inner mitochondrial membrane. All right, let's move on to looking at our driving forces. So here we get to looking more about more at transport, so cellular transport, the structure of the cell membrane, etc. So let's talk about the driving forces for cellular transport, right? So there were some terms that we used, so let's just look at what those symbols and terms mean. So VM, we said, is the membrane potential. And this was basically the unequal distribution of charge across the membrane. All right. We also talked about the equilibrium potential. So the equilibrium potential, which was basically determining the concentration gradient, or determined by the concentration gradient, excuse me. Another thing that we said here was that the equilibrium potential was the membrane potential at which a certain ion was at equilibrium. In other words, its chemical and electrical driving force were equal and opposite, right? So therefore, the potential at which the particle movement is zero. So it's the membrane potential at which the movement of a particle is at zero, right? In other words, the ECDF equals zero. So there's no movement of the particle, okay? We also talk about the CDF, just to review those here as well. This is based on the concentration gradient. So this is the chemical driving force. i.e. the concentration gradient. And then we talked about the EDF, which was the electrical driving force. And that was based on the charge of the particle in question. So this charge of the particle in question. And the polarity of the cell, right? The negative versus positive environments of the intracellular, extracellular fluid. So the polarity of the cell. All right. And then we talked about deciding the ECDF, the overall direction of particle movement. And that was basically the sum total of the CDF plus the EDF. And we looked at that algorithm. So go over that algorithm that we looked at in deciding the overall direction, right? If the individual directions were in the same direction, individual forces, excuse me, were in the same direction, then we simply combine those and the overall direction will follow that. But if they were in different directions, we had to look at the membrane potential, and then we had to look at the equilibrium potential, right? We had to see which value was greater, and then that will determine the direction of the overall force. So just go over that algorithm as well. All right. Let's move on to, actually, let's look at the active versus passive transport. I don't think I have enough space, so clear this. Let's talk about active versus passive transport here. So active versus passive transport. So let's look at this in a tabular format. So I'm just gonna hopefully draw some straight lines here. So let's talk about passive over here, and then active on this side. Now we know that there are subtypes for each of these, right? There are 3 different flavors, so to speak, for passive transport. And then we had 2 main types of active transport. So passive transport, we said, was spontaneous. Another way to say that was downhill. Okay. We can also say that it's going to donate energy. All right. The types of passive transport were simple diffusion, okay. We also had facilitated diffusion, and channels. Okay. Here I'm going to put transporters, otherwise known as carriers. And remember, carriers played a more active role in that transport process than channels, for example. Over in active transport, we said that this was quite the opposite. This is non-spontaneous. All right. Another way to say that is uphill, and then requires energy. Okay. Here we basically had 2 types, primary and secondary active transport. Primary and secondary. Okay, so let's fill in some of the details of each of these. So for simple diffusion, we said that this was based on the concentration gradient. We needed to have a concentration gradient for simple diffusion to occur, right? We also said that the magnitude of the driving force, the surface area exposed, and then the membrane permeability were the factors that affected the rate of simple diffusion. So the factors, let's just try to see your factors. Affecting rate of simple diffusion. Those include the magnitude of the driving force, that is the chemical driving force, in other words, the concentration gradient. The second factor was basically the surface area exposed. And then the third factor was the membrane permeability. Okay. Now, what I want to say here is that we want to distinguish between factors that affect the rate of simple diffusion from the further subdivision of factors that affect the membrane permeability. Remember, we talked about other factors that affect the membrane permeability. Those were, I'm going to list them under here. We talked about temperature being one. We talked about the thickness of the membrane, right? We talked about the size and charge of the particle. Okay. And so these are factors that affect the permeability. they don't necessarily affect the rate of simple diffusion. So let's say we had a thick membrane, that's going to decrease the membrane permeability. But what's going to affect the rate is going to be things like the magnitude of the driving force. That's going to determine how strong the force that wants to move the particle is. That would be affecting the rate of simple diffusion. But in terms of whether or not that thing will get through the membrane, that's the membrane permeability, and that will be affected by things like the thickness, the size of or the charge of the particle. All right, let's see. Someone's asking if the test is open note. No, the test is not open book or open note. You should be studying and prepared to take the test in a fully tested, full testing environment. You shouldn't have any open notes, any open resources. It should be all from your recall, from your memory. Okay, so just to clarify that, you can have scratch paper, so you can draw out tables, if you want to make note of anything during the test, that's fine, but you shouldn't have any open notes to reference. All right, let's talk about facilitated diffusion. So facilitated diffusion, quickly, this is requiring transmembrane proteins. Oops. So here we require transmembrane proteins. All right, which we basically said are carriers was another term here. And this was going to be actively involved. Okay, in that it's actually going to shapeshift and carry that particle across the membrane. The example we listed here was GLUT4, the GLUT4 transporter. Channels, on the other hand, were also transmembrane proteins. In that they have to span the entire membrane. These are also described as passageways or pores. And these examples we talked about, aquaporins, which are the water channels, or we talked about ion channels as another example, right? These are going to be passively involved in transport. And I hesitate to use the word actively here. Remember, this is all still passive transport. When I say actively here, I mean that it's actually intimately changing to help bring this particle across the membrane. It's not going to require energy as such. And so we want to think about its role in actually moving that thing across the membrane and not so much its energy requirement in terms of the type of transport. All right. Next, we look at the types of active transport. These are the types of transport that require energy. In primary transport, the main difference is that there's hydrolysis of ATP. So hydrolysis of ATP, which usually requires a pump, some type of pump is required. And an example was that of that, was the sodium potassium pump. Okay, but the main takeaway is that the pump here has to hydrolyze ATP in order to donate the energy for primary transport. Secondary, on the other hand, energy released from passive transport. So the energy to move things according to secondary active transport is released from passive transport of ions, right? So sodium or potassium moving according to their gradient would help donate the energy. And then the example would be the sodium-linked glucose transporter. Which was an example of co-transport. Right? And then we also talked about the counter transport, which is moving in the opposite direction as well. So that's going to be another example of secondary active transport. So it can be co-transport both items moving in the same direction, or counter transport moving in opposite directions. All right, I'm going to stop here as far as things I'd like to review. These are important concepts. They're going to come in different ways, right? So you'll see them tested in different ways across the questions. So I would start with these topics as far as isolating things to review over the next few days. But then also think about some of the practice questions that we did in class. So think about if you had any difficulty with any of those, go back over those.